everyone, my name is Kinya. I have spent more than 10 years studying goldfish evolution and development. Recently, my friend asked me to make a detailed video about goldfish developmental evolutionary biology. So I have decided to produce this video series based on my papers and my book. We will explore Goldfish EvoDevo. The breeding process of goldfish, many varieties of twin-tailed goldfish were created. When we take a closer look at the caudal fins of these twin-tailed goldfish, we can see that the bifurcated caudal fin consists of left and right duplicated caudal skeletons. Considering that the skeletal structure of a caudal fin is part of the axial skeleton, this means that part of the axial skeleton has bifurcated in twin-tailed goldfish. It is quite difficult to find similar mutations in other vertebrates in the wild. So what's happened to the body of the goldfish? In order to clarify the cause of the bifurcation of the caudal fin of goldfish, we examine the genes of twin-tailed goldfish. Our experiment proceeds as follows. First, we compare the sequence of genes expressed in the embryos obtained from wild type and twin-tailed goldfish. We discovered that the Chordin gene had a peculiar mutation. This mutation was not only found in some twin-tailed goldfish but was observed in the majority of twin-tailed goldfish we examined. Chordin is a crucial gene involved in the embryonic development, specifically in forming the dorsal and ventral side of the body. It suppresses the activity of the gene that creates the ventral side while promoting the formation of dorsal side. This gene is conserved across all of the vertebrate. Such genes which are shared among the species with a common ancestor are called homologous genes. As mentioned in previous episode on this channel, goldfish and zebrafish are closely related, so it is not surprising that this gene is conserved in both. When the Chordin gene function is reduced in zebrafish, most of them die, but some individuals appear with split tail fins. Observation of their embryos revealed that the caudal fin fold split into two. So what about goldfish? Comparing wild type and twin-tail embryos revealed that the embryonic caudal fin membrane is split in twin-tail goldfish. This suggests that in twin-tail goldfish, the mutation in the Chordin gene observed through sequencing analysis causes the caudal fin to split into two. However, it is important to note that the significant difference between goldfish and zebrafish is that the goldfish lineage has undergone genome duplication. In other words, goldfish have two copies of Chordin gene, whereas zebrafish have only one. Since the number of genes can be confused, let me provide a more detailed explanation. In sexually reproducing organisms, including humans, chromosomes are inherited from both parents, one set from the father and one from the mother. Occasionally, chromosomes inherited from fathers and mothers may exhibit slight differences. This means that when comparing the chromosomes inherited from both parents, there are regions that align but contain variation in certain places. This might seem increasingly complicated and confusing, but don't worry. Using terms like locus and allele from genetics can help clarify things. I will use an example involving a father and a mother to explain. Even if father and mother goldfish have slight differences in their genes, they are of the same species, so their genomes largely correspond and can be compared. In this context, specific locations on chromosomes where the same gene is present are called locus, and we can name them for distinction. And when differences are observed at a specific locus between parents, the term allele is used to describe these variations. In goldfish, genome duplication has doubled the number of chromosomes. This raises a question about the relationship between these chromosomes. At least we can distinguish the chromosome region containing the coding genes, confirming they are different loci. As a result, the duplicated coding loci receive different alleles from the father and mother. In zebrafish, it is known that inheriting mutated coding alleles from both parents increases mortality but can result in duplicated caudal fin. Similarly, in goldfish, inheriting mutated alleles from both parents might also cause the twin tail. To confirm this, we conducted a crossbreeding experiment. First, sperm and eggs were collected from a wild type and twin-tail goldfish are fertilized artificially. The hybrid goldfish obtained from these parents were raised to adulthood. Eggs were then collected from these hybrid and fertilized with sperm from twin-tail goldfish to produce next generation. Essentially, we used wild type and twin tail as parents, crossed them to produce the F1 generation, and then backcrossed the hybrids with twin tail goldfish. We categorized these individuals based on whether they resemble wild type or twin tail and analyzed the coding genes in each. The result shows that goldfish that inherit mutated coding alleles from both parents consistently displayed the twin tail phenotype. In the F1 hybrid generation, one mutated allele from the twin tail parent and the wild type allele from the wild type parents were inherited. When the F1 hybrids were crossed with twin-tail goldfish, some individuals inherited mutated alleles from both parents, while others inherit only one. Those inheriting mutations from both parents exhibited twin-tail. This demonstrated that correlation between coding gene mutation and the twin-tail phenotype. So what exactly is the causal relationship here? We conducted some additional experiment to investigate further. First, let me explain the experiment analyzing gene function. This is a rescue experiment. Analysis of the mutant allele of the coding gene revealed the stop codon mutation. This mutation prevents the production of the normal messenger RNA. As a result, fully functional coding protein cannot be synthesized in the twin-tail goldfish embryos. But we can synthesize fully functional messenger RNA in vitro using recent molecular techniques. In this experiment, wild-type messenger RNA was synthesized in vitro and injected into fertilized eggs of twin-tail goldfish. The injected messenger RNA is expected to produce normal coding protein. This would allow embryonic development to proceed like the wild type. As expected, fertilized eggs obtained from twin-tail parents developed into single-tail goldfish embryos, and single-tail goldfish hatched from them. This goldfish grew healthily as a single-tail goldfish. A happy ending, right? Well, not quite. Doesn't something seem strange? When the coding gene is disrupted in zebrafish, most of them die. So why can goldfish survive with a defective coding gene? To uncover this mystery, we need to understand two things. These two coding genes originated from a common ancestor, but their expression patterns seem to be diverged. Using a method called in situ hybridization, which visualizes the location of gene expression. Interestingly, the mutant allele in twin-tail goldfish is associated with a coding gene that exhibits broader expression. As mentioned earlier, the coding gene is known to suppress the ventral gene expression while promoting dorsal side formation. This suggests there might be differences in the ventral gene expression between wild type and twin-tail goldfish. We investigated ventral expressed genes using the same method. Indeed, compared to the wild-type goldfish, twin-tailed individuals exhibited differences in the expression of ventral genes. In twin-tailed goldfish, ventralized expressed genes seems to show abnormal patterns, supporting the hypothesis that the Chordin gene mutation affects the dorsal ventral axis formation and the caudal fin development. So what about the dorsal side? The dorsal side of vertebrate is an important region that gives rise to the neural tube, and it is indeed possible that the reduction in this area could lead to lethality. Let's compare the expression of gene in part of the embryo's brain between the wild type and the twin tail. As a result, we found that there was not much difference between the two. This observation suggests that the duplicated coding genes in goldfish help prevent the lethality observed in zebrafish, allowing twin-tailed goldfish to survive despite mutations in one of the coding genes. As mentioned at the beginning, the mutation of this gene was present in all the twin-tailed goldfish we examined. Moreover, similar results were confirmed in papers published after our research findings were released. Therefore, it is plausible to consider that the mutation of this coding gene was present at the stage of the common ancestor of all twin-tailed goldfish. Breeders could not directly observe the allele with a mutation in the coding gene. However, any goldfish inheriting this mutated coding allele from both parents would have a high probability of displaying some form of mutation in the caudal fin. If the caudal fin was even slightly different from the other goldfish, forming a beautiful twin tail, breeders would likely notice it and select it for breeding. As a result, the mutated allele, which was initially rare among Crucian carp and goldfish, became more common as twin-tailed goldfish were selectively bred by breeders. There are very few vertebrates with bifurcated tail in the natural world. Such organisms mostly appear in fantasy and anime. This is because such morphological mutations require modification of highly conserved developmental mechanisms. Generally, changes in such evolutionary conserved developmental mechanisms are thought to take a long time. However, in the case of goldfish, it is believed that the combination of gene duplication, mutation, and selection occurred at the right time, resulting in a significant morphological change in a very short period. From the perspective of goldfish evolutionary developmental biology, we can see that evolution does not necessarily require a long period of time. Given the right conditions, large-scale morphological evolution can occur relatively quickly, as demonstrated in goldfish. I would be glad if you could understand this concept. It is time to say goodbye. I hope you learned something new about goldfish development and evolution from this episode. Please be sure to subscribe to our channel to catch the next episode. See you soon.